The Neuroscience of Fasting, What Every Muslim Should Know. Fasting, a practice deeply embedded in the Islamic faith, transcends mere abstinence from food and drink. Beyond its spiritual significance, fasting, particularly during Ramadan, triggers a cascade of physiological and neurological changes that are increasingly being understood through the lens of neuroscience. This exploration delves into the intricate relationship between fasting and the brain, offering insights into the cognitive, emotional, and neuroprotective benefits that can be gleaned from this ancient practice. Understanding these neurological underpinnings can provide a deeper appreciation for the holistic benefits of fasting, motivating a more mindful and intentional approach to this pillar of Islam. The initial hours of fasting initiate a metabolic shift. Typically, the brain relies on glucose derived from carbohydrates as its primary energy source. However, as glucose stores deplete during fasting, the body begins to break down stored fat into ketones. These ketones then become an alternative fuel source for the brain. This metabolic switch is not merely a survival mechanism. It has profound implications for brain function. Ketones, particularly beta-hydroxybutyrate (BHB), have been shown to enhance cognitive function, improve memory, and even protect against neurodegenerative diseases. BHB acts as a more efficient fuel than glucose, leading to increased mitochondrial energy production within brain cells. This enhanced energy production can result in improved mental clarity, focus, and overall cognitive performance during fasting. Furthermore, ketones have been shown to increase the production of brain-derived neurotrophic factor, BDNF, a protein crucial for neuronal growth, survival, and synaptic plasticity. BDNF plays a vital role in learning and memory. It acts like fertilizer for the brain, promoting the formation of new connections between neurons and strengthening existing ones. Increased BDNF levels during fasting can enhance cognitive flexibility, making it easier to adapt to new situations and learn new information. This neurotrophic effect of fasting may contribute to the heightened sense of mental acuity and focus often reported by those who fast regularly. Moreover, BDNF has antidepressant effects, potentially mitigating the mood swings and irritability that can sometimes accompany dietary changes. The surge in BDNF during fasting can contribute to a more resilient and adaptable brain, better equipped to handle stress and cognitive challenges. Beyond ketones and BDNF, fasting also influences the levels of various neurotransmitters in the brain. Dopamine, a neurotransmitter associated with reward, motivation, and pleasure, is released during fasting. This release may contribute to the sense of well-being and satisfaction experienced by some individuals during fasting. The release of dopamine could also help to curb cravings for unhealthy foods, making it easier to adhere to the fast. Serotonin, another crucial neurotransmitter involved in mood regulation, sleep, and appetite, is also affected by fasting. While the exact effects of fasting on serotonin levels are complex and can vary depending on individual factors, some studies suggest that intermittent fasting may increase serotonin levels, leading to improved mood and reduced anxiety. These neurotransmitter changes, orchestrated by the metabolic and hormonal shifts induced by fasting, contribute to the overall neurobiological impact of this practice. Inflammation plays a significant role in many neurological disorders, including Alzheimer's disease, Parkinson's disease, and multiple sclerosis. Chronic inflammation can damage brain cells and impair cognitive function. Fasting has been shown to have potent anti-inflammatory effects, both in the body and the brain. During fasting, the body activates autophagy, a cellular process that removes damaged or dysfunctional cells and cellular components. This cellular housekeeping helps to reduce inflammation and promotes cellular health. In the brain, autophagy removes damaged proteins and organelles, preventing the buildup of toxic substances that can contribute to neurodegeneration. Furthermore, fasting can reduce the levels of pro-inflammatory cytokines, signaling molecules that promote inflammation. By reducing inflammation and promoting autophagy, fasting can protect the brain from damage and potentially slow the progression of neurodegenerative diseases. Fasting also impacts the gut microbiome, the complex community of microorganisms that reside in the digestive tract. The gut microbiome has a profound influence on brain health through the gut-brain axis, a bidirectional communication pathway between the gut and the brain. Fasting can alter the composition and function of the gut microbiome, promoting the growth of beneficial bacteria and reducing the abundance of harmful bacteria. These changes in the gut microbiome can influence neurotransmitter production, immune function, and inflammation, all of which can impact brain health. For example, some gut bacteria produce short-chain fatty acids, SCFAs, such as butyrate, which have anti-inflammatory and neuroprotective effects. By modulating the gut microbiome, fasting can indirectly influence brain function and contribute to overall neurological well-being. The potential neuroprotective benefits of fasting are particularly relevant in the context of aging. As we age, the brain becomes more vulnerable to damage from oxidative stress, inflammation, and the accumulation of toxic proteins. Fasting can help to mitigate these age-related changes by promoting autophagy, reducing inflammation, and enhancing mitochondrial function. Studies in animal models have shown that intermittent fasting can extend lifespan and protect against age-related cognitive decline. While more research is needed to confirm these findings in humans, the evidence suggests that fasting may be a valuable tool for promoting healthy brain aging and reducing the risk of neurodegenerative diseases. For Muslims, the practice of fasting during Ramadan is not merely a religious obligation, but also an opportunity to experience these profound neurological benefits. However, it is crucial to approach fasting in a mindful and healthy way. It's important to ensure adequate hydration during non-fasting hours, consume a balanced diet rich in nutrients, and avoid overeating during iftar and suhoor. Individuals with pre-existing medical conditions, such as diabetes or epilepsy, should consult with their healthcare provider before fasting. In conclusion, the neuroscience of fasting reveals a complex interplay between metabolic, hormonal, and neural processes. Fasting triggers a cascade of beneficial changes in the brain, including enhanced cognitive function, improved mood, reduced inflammation, and neuroprotection. By understanding these neurological underpinnings, Muslims can gain a deeper appreciation for the holistic benefits of fasting and approach this sacred practice with greater intention and awareness. This knowledge empowers individuals to optimize their fasting experience, reaping not only the spiritual rewards, but also the tangible benefits for brain health and overall well-being. Further research will undoubtedly continue to unravel the intricate relationship between fasting and the brain, providing even more insights into the transformative potential of this ancient practice. Embracing the wisdom of fasting, guided by both faith and scientific understanding, can pave the way for a healthier, more resilient, and more fulfilling life.